Hi everyone, we are looking at part 2 of chapter 12 in this lecture. In part 1 we discussed this guy named Johann Gregor Mendel, and specifically we talked about his experiments on pea plants and all the different traits that pea plants have and how they are passed to their offspring. Well, we're going to talk a little bit more, we're going to summarize a little bit more about the laws that Mendel came up with after all of his experiments, and then we're going to go ahead and talk about how some of these laws are violated. In reality, the way that traits are inherited does not just simply follow the rules that Mendel came up with about pea plants, but it actually can be quite a diverse array of inheritance patterns that differ from the ratios we would suspect given Mendel's laws of inheritance. All right, so here are Mendel's laws, and most of them we've already kind of subtly talked about. We just didn't call them a law. His first law was that there's the law of dominance, meaning if there's two alleles, one that's dominant and one that's recessive, that the dominant allele will be the one that is shown phenotypically on the organism, and the recessive one will be masked. So in our infamous example of hair color, where brown hair is the dominant allele and blonde hair is the recessive allele, well, if we were to denote these on an allele key, big B we would say is, or brown hair we would say is big B, blonde hair we would say is little b, recessive, dominant. Okay. What this means is that, let's say an individual is a heterozygote, meaning they had both a copy of their one chromosome bared the dominant allele and the other chromosome bared the recessive allele, so they were big B, little b. What this law of dominance says that this individual, the dominant allele is the one that will be shown and the recessive one will be masked or concealed. So this individual would have brown hair, they wouldn't have blonde hair, nor would they have some sort of intermediate hair color in between dominant and recessive. They would just have the dominant phenotype, the recessive one would be pretty much concealed. So that's what his law of dominance stated. Equal segregation of alleles. Basically, there's always two alleles, because we have got two sets of genes, one from our mom, one from our dad, that we're going to have two different alleles. And basically, equal segregation of alleles means, when the person has two copies of an allele and they're making gametes, that the gametes will have a 50-50 likelihood of inheriting either allele, and it's just going to be one allele is just as likely to be inherited as another. So, for example, in this scenario that we said maybe somebody had just for the sake of understanding here, let's say we had a heterozygous person that had one trait for the dominant allele, or they had a dominant allele and a recessive allele. Well, when this person goes to make gametes, this would represent a diploid scenario where the person has got two genes, one that they inherited from their mom and one that they inherited from their dad. And when they go to make haploid gametes, only one of these is coming with. Okay, and so whether it's the little b or whether it's the big B, it's just as likely that the little b could come as it is that the big B could be packaged up into the gamete. All right, so that's kind of what equal segregation of alleles mean. It's not like somebody with dominant hair. The dominant allele is much more likely to be packaged into the gamete versus the recessive one. No, for any two alleles that are on homologous pairs of chromosomes, either one is just as likely to be passed down into a gamete. His last rule was that independent assortment, inheriting one gene does not influence the likelihood of inheriting another gene. So, for example, let's say that there's two traits, maybe let's say seed texture and flower color. Well, it's not the case that, like, if let's say some offspring from a plant inherits purple flowers, that they're going to be much more likely to have a smooth seed texture versus wrinkled seed texture. These two traits are inherited separately. That there's a 50-50 shot of getting either allele for flower color, and that does not affect the probability of getting the alleles for seed texture. Traits don't influence or change the likelihood of kind of this equal segregation. All right, so those are his laws. Now we're going to talk about some alternative inheritance patterns, though, and how they contrast those laws that Mendel came up with. The first are going to deal with his law of dominance. Okay, so there's a couple of ways where this law can actually not be true, and here's what they are. The first is called incomplete dominance. And this is a scenario in which there is actually a heterozygote that has the phenotype that's a hybrid of the two parental phenotypes, either dominant or recessive. So in this picture, an example of incomplete dominance you see here, this plant is homozygous recessive and they have white flower petals. This plant up here is homozygous dominant and they have red flower petals. And if you were to cross one of these homozygous dominant plants with a homozygous recessive plant, you would get a whole bunch of offspring that are heterozygotes, meaning they have one allele from the dominant parent they received from the parent with the dominant phenotype, and one allele that they received from the parent with the recessive phenotype. Okay. And note here, though, that the heterozygotes, their phenotype, meaning their appearance, their phenotype is like pink, and that's kind of halfway between red and white. Okay, so this is an example where the offspring have a blended phenotype, one that's like kind of a hybridization or a merger of the two phenotypes above. And this was the very type of idea that was predominant in the 1800s during Mendel's day, and he actually was making the case that this does not happen, but he was a little bit short-sighted. In reality, this does happen. It's just a little bit more of a violation to the rule as opposed to the norm of the rule. Okay. So if that does happen, it is called incomplete dominance, and it violates Mendel's law of dominance. Okay. Another type of scenario could be codominance. In this situation, what you see is two dominant phenotypes expressed simultaneously on an organism. So this happens in cows. So you might have a cow that has white color. Well, white is in fact a dominant allele. But there's another dominant allele for kind of a red or brown color. Okay, so this is also capitalized. It's a different dominant allele. Well, when you take this dominant allele from this cow and cross it with the dominant alleles in this cow and end up with a cow that's got inherited one dominant allele from this parent and one dominant allele from this parent, now they have both dominant alleles and they actually are showing both phenotypes. They're showing a little bit of the brownish reddish color from this parent, and they're showing a little bit of the white color from this parent. So two dominant phenotypes expressed simultaneously. That is called codominance, and that again violates this law of dominance, that there's only going to be one dominant trait and the other one must be recessive and is masked in the presence of a dominant allele. Here we have two dominant alleles both being shown. There's another scenario where we could have multiple alleles. In reality, oftentimes there's not just two alleles, a dominant and a recessive allele. Sometimes there can be a variety of alleles, and a good example of this is blood type. So the allele key generally that we use for blood type is IA, IB, and I. So these two are dominant alleles, as indicated by the fact that they're capitalized. This one is not capitalized, so this would be a recessive allele. So here we actually have three alleles, two dominant and one recessive. And this corresponds to different blood types. So somebody that would have either IA, IA, that would be homozygous, or it could be IA and little, oops, sorry, my battery's running low, or little I, would both have the blood type A. Okay, so either one of these, the heterozygote or the homozygous dominant, would have blood type A, or similarly, if somebody had IB and IB, so homozygous, or IB and little I, the heterozygote, both of these would have the same phenotype. We'd call that blood type B. Okay. Blood type is also a pretty interesting example of codominance, because if somebody inherited this dominant allele and this dominant allele, you know, they inherited this one from one parent and this one from another parent, they would have IB, IA, and their blood type would actually be AB. Okay, so both the dominant alleles, the B and the IA, are being expressed, and that would make type AB blood. So there can be multiple alleles, and then again, this is also an example of codominance. Generally, when there's a whole bunch of alleles, it's not as clearly defined which one is dominant and which one is recessive. So generally, the convention used here in this scenario is that the most common allele we call wild type, and then all of the other ones that are different from that we call variants. So there can be an example of like mouse fur color is the one that's in the book, I believe. And mice can have all sorts of different hair color. It can be white, it could be brown, it could be black, it could be gray, it could be spotted or not. There's a lot of different fur colors out there, and so because of that, there's a lot of different alleles for fur color. There's the brown fur allele, the white fur allele, the black fur allele, the gray fur allele. And in this scenario, you might wonder, well, which one is dominant and which one is recessive? And then basically you would, in order to make that determination, have to survey the population, figure out which one is the most common, and then that is just the one that you would simply call the wild type allele. And usually it's denoted with a plus sign on an allele key. So let's say that brown fur was the, let's say you're making an allele key and there was BR stood for brown fur, white stood for white fur, B stood for black, and maybe gray for gray fur, and this was your allele key. The wild type one, if it was, let's say, brown, you'd put a little positive or plus sign there to indicate that this is the wild type allele. And then what we would say then, meaning this is the one that's like most commonly in nature, and then these are kind of the ones that are going to be different than that, and so we call these all these other types of alleles variants. Okay. So not always do we just have two alleles, a dominant and a recessive. Sometimes there's a variety of them. Okay, one last thing then here, linked genes. This is what's really going to violate this one here, independent assortment. Okay, and to understand how linked genes work, let's go back to this old review question that we had on part 1 of this chapter 12 lecture. This was the review question. We had two plants crossed together, and they had two traits each. One was for seed texture was the one trait, seed color was the other trait. In this plant, it was a heterozygote for seed texture, meaning it had one allele for round and one allele for wrinkled, so big R and little r. And then for the trait of seed color, it was also a heterozygote. It had one allele for yellow and one allele for green. Okay, so two heterozygous combinations there for both of those traits. Then this plant was a heterozygote for seed texture because it had one allele of the dominant allele and one allele of the recessive allele. And then for seed color, it was homozygous recessive. It had two alleles of the recessive allele, which in this case is green colored seeds. Okay. Well, when we talked about how you would do a Punnett square analysis in this cross, we basically had the assumption that these traits will sort independently from one another. So let's say when gametes are being produced from this plant, it's going to take one of two of these and one of two of these and package them in together. So here's the different combinations. It could take the first of each, package them together, or it could take the first of one and the second of the other, and that would get packaged together, so that would be big R little y. Or it could take the or it could take the second of the first pair and the first of the second pair, so the little r, big Y. Or it could take this and this, the second of each pair, little r and little y. So the way that the r's and the y's sort out, that's equal and it's independent. Well, that assumes that these are on different chromosomes. So let's say if seed texture was on chromosome number 1, okay, and let's say that this was the big R and this is the little r, and let's say the genetic information for seed color was on chromosome number 3, we would have a big Y and a little y. Then when these chromosomes are sorting out into the gametes, you know, this one could go here, and then the one that also goes in here could either be this chromosome or it could be this chromosome. Okay, so they could sort out independently from one another. This one could go to join this one, or maybe this one could join. It's all up in the air. But what if the traits were on the same chromosome? So here this was, let's say, on chromosome number 1, both the seed texture and the seed color were on the same chromosome. Well, if that's the case, then basically all of these, you know, all of these would always be passed on together when they're going to make the gametes. So these would not independently assort. So the big idea is that linked genes, we would say, first and foremost, that's multiple traits on the same chromosome. And because of that fact, they don't assort independently, that they are inherited together, if that makes sense. Okay. All right, so that is another thing that can happen, linked genes. And then the last one here, the X-linked inheritance pattern. This is going to be, we talked a little bit about this already, but when we're talking about X-linked traits, it's a little bit different because men only have one X chromosome, an X and a Y, and women have two X chromosomes, two X's. So if there is a trait that's on the X, then men are going to probably be a little bit less likely to have any choice in the matter of what's inherited, because they're just going to inherit it, the X. Whereas women, if there's a trait on an X chromosome, they could either have the dominant or the recessive. It doesn't matter. Whereas men, if they have the dominant or the recessive, they're just going to have that dominant or recessive phenotype, because the Y doesn't really contribute much in this scenario. So these can be a little bit different. Here's kind of a picture. Let's say that there's a dominant allele on the X chromosome and a man has it. So he's got an X and a Y, and on the X is the dominant allele. And then let's say there's a woman who has an X-linked recessive allele, which is the red one, as well as a dominant allele. Well, when they have offspring, these are all the combinations that could happen. Okay, this X could go here or here, and then this X goes here. Or oops, my bad again. This one, the Y goes here, and the Y goes here, and this X goes here, and it also goes over here. Okay, and then these get sorted out. This is a kind of a convoluted picture. I don't really like this, but basically all that to say, when you look at all of the phenotypes of the offspring, and you're looking at which offspring are going to show this X-linked recessive trait, it's only the son, really. The rest of these are unaffected, and that's because, you know, in this scenario, this son inherited the unaffected X, which was good. And then in this scenario here, one of the daughters, they inherited this X and then also this X. In this scenario here, they just inherited this one and then the Y. And so they will have whatever phenotype is the recessive one here. And then this combination here, the other daughter, they actually do inherit the recessive trait, but they also inherit, because they have two X's, they also inherit the dominant one from dad, and then that means that they have the dominant allele there, so it's going to mask or cover up the unaffected, or cover up the recessive allele here. So all that to say, there's generally a greater likelihood that sons will inherit an X-linked recessive trait versus daughters. And there's a whole bunch of X-linked disorders out there, like color blindness is one that I think is X-linked. The disorder Duchenne muscular dystrophy is X-linked, so that's going to be much more common in boys. And I believe there are some others as well. All right, so I guess that's a good segue here, just genetic disorders. There are a couple of different types of disorders. We would say, you know, genetic disorders are a little bit different than a disease. A disorder is something that's inherited and can't really be done away with. We're talking about something that's in the genes. It's hard to go in and change somebody's genes or fix genes. Now they are working on a way to do that, but up to this point, it's pretty much impossible. So if one is born with a genetic disorder, it's likely that they will have that disorder for the rest of their life. That's different than a disease, which could be like a bacterial or a viral infection that can come and go. Well, when we're talking about genetic disorders, there are some that are recessive genetic disorders, and what this means is that both copies of the allele must be recessive. So in order to actually have the disorder, the individual must be homozygous recessive. Okay, and an example of this is albinism. So to be albino, the albino phenotype means there's a lack of melanin made, which is the pigment that makes your hair darker or your skin darker or lighter. So more melanin means more darkness of the skin, and so if there is no melanin, people with albinism have very light skin, and kind of even can be like a blondish or a whitish hair color. And that is a mutation in the chromosome that produces melanin, and in order to have that phenotype, one must inherit both alleles that are recessive. That's not usually the most common genetic disorder. The more common ones that are out there in the world are the dominant genetic disorders. And I guess I should take that back. They're not the most common. If the dominant genetic disorder is a really severe one, then they're actually quite not common at all. They're more rare. But if the dominant genetic disorder is the symptoms aren't very severe, then it would be more common. Okay, an example of this would be Huntington disease. So somebody that inherits just one chromosome that contains the Huntington disease allele, they would have this genetic disorder. They don't need to have two recessive alleles to have it, only just the one dominant, and then that will be the one that is phenotypically expressed or shown by the organism. All right, another topic here, pedigree analysis. This is sometimes a tool that genetic counselors will use to just determine how a genetic disorder is flowing into and out of a family. And given if there's some genetic disorder that runs in somebody's family, they may be concerned about if they'll pass that genetic trait, that genetic mutation or disorder to their offspring. And so they could consult a genetic counselor who can do a pedigree analysis and kind of see what the likelihood of passing that trait on would be. So this is an example of what it looks like. Basically, the squares represent males, and the circles represent females. And then also blue means that the individual is affected, and then this yellowish brownish color means unaffected. And specifically, this is an analysis for a disorder called alkaptonuria. And I'm not too familiar with this disorder, but it is autosomal recessive, meaning it takes two recessive alleles in order for the individual to have that disorder. By the way, this word autosomal, this means chromosomes that are not sex chromosomes. So this would be anything that's on a chromosome number 1 through 22. The 23rd pair of chromosomes is the sex chromosomes, so those would not be autosomal. So this disorder, alkaptonuria, if that's how it's pronounced, is autosomal, meaning it requires two recessive alleles, and those alleles would be some chromosome between this number of chromosomes. It's not one of the sex chromosomes. All right. In either case, we can see here that the first the father of the first generation and the mother of the first generation. The mother did not have the disorder, but the father did. So if we know that the father had the disorder, my question to you is: knowing that it's autosomal recessive disorder, could we figure out what genotype this individual is? And the answer is yes. Okay, we know that in an autosomal recessive disorder, the person must have both the recessive alleles. Well, in this scenario, it looks like the dominant recessive alleles are expressed as capital A and little a for alkaptonuria. So, if we have a recessive disorder, that means this individual must have two copies of the recessive allele. Okay. And then we're wondering, what about, maybe we're looking back and wondering if perhaps the mom of this first generation had any alleles that were the recessive allele, and you would be able to figure out, yeah, she did, because if you looked at the kids, you see that this son had the disorder. The only way you can have the disorder is to have two copies of the recessive allele, and you inherit one copy from each parent, so that means that this person had inherited one from dad, and then mom must have had one to provide as well. So we know that mom in the first generation is heterozygous. She has one dominant allele and one recessive allele for this disorder. And we know that dad is capital A, capital A. Okay, and then Similarly over here, this is the same story. We're wondering, let's say that some man marries into this family, and by the way, when you're looking at a pedigree like this, basically what this is showing is that this is the son that this couple had, and then there was some woman that had kids with that son, and so these two had these 3 kids, and then this person married, or had some kids with that person. This person had some kids with this person, and here's their child, and here are their childs. So that's how these are read. And then you basically, the long and the short of it is you're able to track how the alleles are flowing through the family tree. And so you could ask the question, what's the likelihood that these parents will have offspring with the disorder, or you could ask what's the likelihood these parents will have offspring with the disorder, or what's the likelihood here. So that's kind of an important tool that some genetic counselors might use. It's called a pedigree. If something like this was on exam, could you tell me what these genotypes were if you didn't know? Just based upon what the offspring have and whether or not the individuals that were in the parents had the disorder. That might be something that you want to be able to do. One last note here on our chapter 12 is this thing called epistasis. And epistasis basically refers to the circumstance in which one gene is going to mask or interfere with the expression of another. So let's say here's a diploid, the chromosomes of a diploid organism. So this would be the chromosomes inside their nucleus. It could be that, let's say, one trait for, let's say, height, maybe, I don't know, maybe it's on chromosome number 1. No, I don't know if it is or not. But epistasis means that there's some gene on one chromosome that can actually interfere with how another trait is expressed. So maybe that this gene here makes some sort of product that then goes and actually shuts off or turns on a gene that's on a different chromosome. So you can have all these sorts of interactions too, creating kind of the genetic landscape of an individual and their phenotypes, because you can have chromosomes actually affecting the expression of other chromosomes and whether they do kind of what they're supposed to or not. So when these types of interactions occur, we call it epistasis. Okay, one last review question here. List and describe the 5 ways in which inheritance patterns can deviate from Mendel's original laws of inheritance. Go ahead and pause the video and work on this for a second, and then I'll go through the answers with you. All right, here are the answers. The first is, see if you can just list them. The first was incomplete dominance we talked about. Then we talked about this thing called codominance. And then we also talked about multiple alleles. We talked about linked genes. We also talked about X-linked traits. Okay, and a quick description of these. Incomplete dominance means that a heterozygote will display a phenotype that's somewhere in the middle between the dominant and the recessive phenotype. Codominance means that 2 dominant phenotypes are shown simultaneously, and that goes contrary to Mendel's law that says that dominant will there'll only be 1 dominant that is shown, and then in like a heterozygote the dominant is shown and the recessive is masked. Well, in this case 2 dominants are shown. Multiple alleles, this means that rather than just being 1 recessive allele and 1 dominant allele, there could be a variety of alleles out there, and they're not necessarily dominant or recessive per se, but in this scenario we can talk a little bit more meaningfully about them by denoting the most common allele with this word wild type. And then lastly, or second to lastly, linked genes. This is where we have 2 traits that are on the same chromosome, and because they're on the same chromosome, when a meiosis occurs and the individual is making gametes, the two genes are inherited or passed down to the offspring together. So let's say, for example, hair color and eye color on the same chromosome. If I passed my DNA to my offspring and they inherited my blonde hair gene, they would also inherit my blue eyes gene because they're on the same chromosome. Okay. It's not like they could inherit my blonde hair gene and then not my blue eye color gene, because when they are passed into the gametes, they're passed as one together. Okay, X-linked traits, again, this is just when there's a trait on the X chromosome, and due to the fact that men and women have a different number of X chromosomes, that can kind of change the inheritance pattern there. All right, that's going to wrap it up for Chapter 12.